Pancreas is an exocrine and endocrine gland. Pancreas originates from endoderm of the germinal layer. Location of pancreas is inferior to stomach. Simply we can say in the band of duodenum, pancreas is located. So, this is the pancreas. In the pancreas, acinar cells and islets of Langerhans is present. These acinar cells are responsible for the secretion of pancreatic juice. With the help of pancreatic duct, this pancreatic juice is transferred towards the duodenum. And as the gland which passes its secretion towards the other gland or other side is the exocrine gland, as it possesses pancreatic duct. This islet of Langerhans are nearly 1 to 2 million in the pancreas, having alpha cells, beta cell, delta cells and pancreatic polypeptide cell, or it is also called as an F cell. This alpha cell is mainly responsible for secretion of glucagon, that is anti-insulin hormone. Beta cell is mainly responsible for the secretion of insulin hormone. Delta cell concerned with secretion of somatostatin, and that is anti-growth hormone, we can say. Pancreatic polypeptide or F cell concerned with the secretion of pancreatic polypeptide hormone. It concerned with the inhibition of secretion of other enzymes from the alimentary canal. In the internal structure of pancreas, 1 to 2% region is occupied by the islet of Langerhans, in which 1 to 2 million islet of Langerhans are present. Here, alpha cell constitute the 15% region of the islet of Langerhans, beta cells are always more, that is 65%, and delta cells are nearly 7% of the islet of Langerhans. Now, if we talk about the glucagon, glucagon is a hyperglycemic hormone, as it concerns with increase in the glucose concentration in the blood. It converts glycogen to glucose, and that's why we can say glucagon mainly concerned with the two processes, that is glycogenolysis as well gluconeogenesis. Glycogenolysis means that is a breakdown of glycogen to increase glucose in the blood. As glycogen is a polysaccharide, it broken down into the monomer called glucose. Second, gluconeogenesis is also induced by the glucagon in the liver cell. Remember that in the liver, from any other organic compound, the carbohydrate, mainly glucose, is synthesized. That's why we use the word genesis over here. Whenever the new word is that, it means from any other organic compound, carbohydrate is formed. And that's why it is called gluconeogenesis, in which from protein or lipid, glucose is synthesized inside the liver. Let's talk about the insulin, which is hypoglycemic hormone, as it is antagonistic to the glucagon. Antagonistic means insulin and glucagon both act on the glucose, but glucagon convert glycogen into glucose, so glucose increases. Insulin exactly opposite in its function, that it always reduces the glucose concentration from the blood. Insulin always induces liver cell, adipose tissue and the muscle cells as well to absorb more and more glucose from the blood. In this case, in the blood, glucose concentration goes on decreasing. Insulin is anabolic hormone. It always induces synthesis of organic compounds like protein inside the cell. So amino acids are used up. Insulin concerned with the glycogenesis. As I told earlier, genesis means formation. It induces formation of glycogen by converting glucose from the blood into the glycogen. So in this case, the monomer get transformed into the polymer unit, and that's why glucose decreases inside the blood. This way, insulin and glucagon concerned with the maintenance of the glucose in the blood. When require, glucose is decreased by the insulin. When require, glucose is increased by the glucagon. Both insulin and glucagon are peptide in nature. The delta cells concerned with the secretion of somatostatin. Remember that this somatostatin is also secreted from the hypothalamus. It is anti-growth hormone. It reduces the secretion of growth hormone and reduce or inhibit the growth of individual. Pancreatic polypeptide cell or the F cell are concerned with the secretion of pancreatic polypeptide hormone. This hormone inhibit the secretion of hormones from the alimentary canal. Alloxan and cobalt chloride test is carried out to understand the physiology of pancreas and simply islet of Langerhans. Alloxan mainly destroy beta cells in the condition when we try to understand the function or the significance of alpha cell as well. Cobalt chloride destroy alpha cells in the same condition when we try to understand the contrary function of the beta cells. Thank you.